Somayeh Dodge is an assistant professor in spatial data science and the director of the Move Lab at UC Santa Barbara. We invited Somayeh to share her experience in dynamic movement data visualization and modeling in both human and natural systems, which have made important contributions to our ability to model complex issues like human-wildlife conflicts, environmental change, natural hazards, spread of diseases, mobility, and global migration. So we're really excited to hear from you and learn more about your work and hear what your visions for the future are. Do you want to take over now?
Sure. Do you see the presentation or the presenter? Okay.
Presentation, it's perfect.
Okay, well, thanks so much for having me, and thanks, Stephanie, for the nice introduction. I'm glad that Sara went before and basically covered most of the things so I don't have to go into depth about the general picture, but overall, so my name is Somayeh Dodge, and I'm at UC Santa Barbara, and I work at the intersection of movement analytics in both human systems and ecological systems. I should admit my background is geographic information science and spatial data science. So I'm not an ecologist, but I work with ecologists. And in my work, I basically do a data-driven analysis and modeling, and also here I specifically would like to advocate for a human-centered approach towards converging human mobility analytics and animal ecology methods. So as we all know, both humans and animals are being tracked everywhere, and we can use these signals to understand the behavior of both animals and humans and their interaction with their environment and with one another in these complex systems. In spatial data science or movement data science, we basically start with the data to develop data-driven analytics and machine learning techniques to understand behavior of individual, understand movement. So going from data to behavior and use this knowledge to improve our modelings and maybe even add to theory. Here, collaboration between data owners, domain scientists, and method developers is key to develop successful and meaningful methods that can be applied to both domains. So this framework, in this framework, visualization and visual analytic tools could be very helpful in interdisciplinary collaboration to bring together scientists and basically communicate patterns that are hidden in the data. The success of this framework depends on how it can enable humans for working with these tools with AI and machine learning to for a better knowledge discovery and more meaningful knowledge discovery. Basically, the tools that we develop should facilitate disciplinary collaboration and optimize human input in the process. And only us as the analyst or the human in the loop can control for the bias in the data and look at how we can control the bias in our algorithms. And also other things that we need to be mindful about. Sorry, this doesn't work, it seems. It's basically creating tools that encourages and facilitates collaboration and communication by taking advantage of visualization. So through this presentation, I would like to show some of my work that basically follows this framework and applies develop methods that bring in human in the picture, and also the methods could be applied to both domains of human mobility and movement analytics. So first of all, in my group, we realized that we need to first create a tool that we can map the data to understand or discover patterns, and maybe even use it as a hypothesis generation tool, and basically interact with the data. So we developed this open source exploratory data visualization tool called Dynamovis, which is available through MoveMang website, and also we publish it in Movement Ecology. So the tool is flexible enough that you can use your tracking data and map it the way you want by changing the variables. So Sara mentioned additional factors that, like environmental variables, that you want to explore the associations with movement to movement. So basically you can map them using different visual variables of color, size of the tracking data, or shape of the tracking data. Next, I will give you an example that captures that better. But in general, the tool gives you flexibility without any need for programming to map data both in 2D and in 3D, although the 3D is not yet released, and map environmental variable over your tracking data. So as an example here, I show a visualization that animates tracking data of albatrosses, Galapagos albatrosses that they forage at the coast of Peru, and their nest is in Galapagos. So here the size of the dots shows how fast the bird was moving or flying, and the greenness shows how, the amount of food availability captured by OceanNet primary production. So as you can see, you're using visual variables size and color. We are capturing the association between speed of the movement and the greenness of the ocean, or food availability in the ocean. And to capture how this movement is assisted by wind, here this visualization uses a visual variable line with different sizes showing different speed. Narrow lines shows slower speed, and thicker lines show faster speed, and their color shows how much wind was helping this bird. So the albatrosses strain into headwind is captured by narrower red lines, and when they are helped by wind, you see blue and thicker lines, and that shows that on the flight after foraging back to Galapagos, these birds use wind assessment. So next, I would like to focus on interaction analysis. So there have been a lot of methods to capture interaction in moving individuals, or in dynamic systems. In geography, we focus a lot on spatial interaction, how individuals interact in space, and but also dynamic interaction, meaning that over time, how individuals interact with each other. This animation shows tracking data of two tigers in both the geographic projection, 2D, the two-dimensional projection, and 3D, the space-time cube. So the time goes vertically upward. So as you can see, these two tigers come sometimes close to each other, and sometimes they move parallel to each other. So we were interested to look at not only concurrent interactions, but which means that they moved closely at the same time in space, and delayed. We wanted to also look at delayed interaction, so which is basically if the individuals visited the same location with a time lag, or followed each other's path with a time lag. So the issue of a scale that Sara nicely mentioned, talked about the general aspect of a scale. Here, I want to focus only on resolution, the problem of resolution and mismatch of resolution in the data set. So when we capture movement at different resolutions, the sampling rate that we use to capture movement impacts our analysis. So for example, here I'm showing that how movement speed is impacted by changing the sampling interval from 3 seconds to all the way to 1800 seconds. And although this is not animal movement, human movement, but that also is generalizable to animal movement. And the path tortuosity is also very much impacted by the resolution we capture our data. So another important aspect of mismatch of resolution or using coarse resolution to capture movement is that you might miss interaction points. Like when the two animals come into close contact, we might miss them if we have a mismatch of resolution or a coarse resolution. So to somewhat help this problem, we developed a time geographic approach to interaction analysis. So in time geography, which was this framework was first introduced by Hagstrand, you model the locations that are accessible to an individual during a time budget with an ellipse, or basically if you consider the space-time path, the space-time cube, the areas that are accessible in space and time to an individual is modeled using the time geography framework, which indicates that, for example, if you have two tracking data, like at time A and B, two tracking or sampling points, the areas that are accessible to the individual between the two sampling points based on the speed of the individual and the time of these two sampling points, what is the accessible location to this area, which then can be used to identify potential areas for interaction. So we call that accessible area a potential path area. So if we compute the potential path area along the tracking data of different individuals and intersect them together, then we can identify areas for potential areas for interaction. And then when we put them in sequences and compute the duration of continuously intersected areas, then we can identify or compute the duration of interaction. So in this paper, we developed a method and applied it to both human movement data and animal tracking data. So here you see the outcomes applied on 3 tracking data of 2 tigers. The home range is shown red and green, and the blue shows a leopard that shares home range with these 2 tigers. And this tracking data is obtained in Western Forest Complex in Thailand through my collaborator, David Smith. So we looked at not only the intersection between their potential path areas, but also the delay that we could detect in the intersection. So like, for example, a tiger goes to a location, and with a delay, a leopard or another tiger goes to the same location. And if you notice here, some of the tracking data has different resolution. So parts of the tracking data is 20 minutes resolution, and parts of it is like 1 hour resolution. And basically, if you look at the delay in interaction, you see that the leopard tends to stay away from both tigers, but shares come close to contact with the green tiger often, because the home range of the leopard is completely within the home range of the tiger. And then the 2 tigers basically have a delay interaction starting from about 3 hours that takes about up to 2 weeks. They go and check out the location of the other tiger, that perhaps for scent marking. So, and if you look at the duration of interaction computed with these two different methods, so here we are using human movement data because we had the travel survey data that we could validate the duration of interaction when people were moving together and stayed together, and then people just interacting together. So we had information about that. So the potential path area, as you can see here, by changing the resolution, at the 1 minute sampling interval, it's fairly comparable to what you get from the diary data or travel surveys. But the proximity-based approach, which is basically computing point-to-point distances between the two trajectories, fails to capture a lot of this interaction. And when you make the sampling rate coarser, proximity-based approach becomes completely invalid, and the potential path area overestimates the duration of contact because the size of ellipses become larger. So, and the other graph, the grayscale graph, shows where if the individuals were moving together or individuals were randomly coming to contact together. So individuals within the same household and individuals outside household. So individuals within the same households were tracked together. You can see those basically with darker gray, and individuals outside household are lighter gray. So for individuals who weren't tracked together, we can see that the proximity-based approach even cannot detect any interaction or contact. However, the potential path area is capable of doing so, but the numbers are overestimated when the sampling interval increases. So to end this talk, I would like to shift the focus towards COVID and capturing human movement throughout the COVID pandemic. So starting with the pandemic, a lot of companies started to share their data. As you can see here, they come in different formats, different resolution, both spatially and temporally. Some of them are freely available, some of them are commercial data sets. So these data have been used a lot in not only modeling the spread of COVID, but also looking at the impact of non-pharmaceutical intervention for ordering shelter in place or limiting mobility throughout the COVID-19 pandemic. But also, as you know, the community, the movement ecology community started using human mobility as an indicator of, or as the two corresponded with a human animal activity to look at the relationship between the two, whether they impact movement of each other in terms of increasing activity or lowering activity, less activity due to activity of the other. A lot of papers that were used for focused on looking at human movement impact in response to COVID, they mainly use one or two or multiple sources of this data. Here we wanted to show, first of all, we wanted to look at how representative these data sets are in United States. We only focused in the United States, and then also looking at different patterns that we identified with this data set. So here you can see we are correlating the population at each county with the user base of some of these data sets, because not all share the number of their user base. So only PlaceIQ, the Scarlet Lab, SafeGraph, and Cubic share their user base. So as you can see here, the population, so we would expect that all these dots, which each one represent one county in the United States, would line up in the regression line. However, in larger metro areas, in counties that represent larger metro area, we see an underrepresentation, meaning that this mobility data do not capture the exact mobility of the population. They might not capture the exact mobility of the population well in larger metro areas. Next, we looked at the spatial and temporal coverage. Here, the color, the intensity of the color shows if we had complete data for an entire year. I think here was 2020. In all counties and in all calendar dates. So as you can see, so on the left, you see the calendar view, and on the right, you see the geographic view. So there are a lot of missing data and gaps in the data set. So if you are using Google data or Facebook data, you might not be capturing the entire mobility throughout the space, and both spatially and temporally. And finally, we were interested to look at what patterns we can identify with this data set. So we applied local indicators of a spatial association to look at clusters, identify clusters. So as this animation moves forward, I know it's very difficult to see the patterns, but we are using two different data sets, Cubic as a commercial source and SafeGraph as an open source data that you can get for free. The hotspots represent high values where closer to, high values are clustered, like counties that have high values near to other counties that have high values, and cold spots are the other way. So lower values of mobility clustered with other lower values. So if you look at these two graphs, like on the left, you see mobility, like travel distance. So these are distance-based measured, and on the right, you see percentage-based measures, which are like the percentage of population sheltered in place, stayed at home. So we see some hotspot and cold spot that during the year overlap, but also a lot of discrepancies and differences between the two data sets, and also between the two indicators as well. So to summarize these patterns, because the animation is very hard to detect patterns, we looked at an aggregated view of these data sets. So each dot represents a county, and the transparency of the color shows whether they were more recent cold spots and hot spots, and older cold spot and hot spots, meaning that at the beginning of the 2020 or at the end of the year. And then the size of the dots represent the number of weeks a county stayed as a hot spot and cold spot. If you just look at this aggregated view, which we actually developed a tool for representing these structures, so you can go to the GitHub page and play with the tool. You can zoom in different counties and in different states, and look at variation in different states as well. So if you, yes.
Sorry, just to jump in, we're running quite over time here. So
sorry.
I want to jump ahead to the
to summarize, the patterns that we get are very different in these two data sets. And with that, I would like to thank you.
Sorry to wrap.
No worries. My computer shows 8 minutes, so I thought I have 2 more minutes for some reason.